Welcome back, my students. Dr. Oogway here, and today I bring you the cardiovascular system from Guyton. And before starting the video, I will tell you the notes of this lecture are in the description of this video. So this is the cardiovascular physiology. It has many chapters, and we are going to start with its first chapter, that is organization of the cardiovascular system. This chapter, organization of the cardiovascular system, is explained by Guyton in 3 learning objectives. If we study these learning objectives, this organization of the cardiovascular system is clear to us. So in this video, we are going to study the first learning objective, that is describe the characteristics of different blood vessels that make up the circulatory system. So this is our question of interest. Describe the characteristics of different blood vessels that make up the circulatory system. The way I teach you this video, it is in this manner you will write the answer of this question in your university examinations, and the notes of this video is in the description of the video. Clearly? So the different blood vessels that make up the circulatory system are, number first, arteries. In arteries, you have to remember 3 points. The 3 points are: they transport blood under high pressure to tissues. Arteries have strong vessel walls and blood flows at high velocity in arteries. Number second point. Number third point, they are also known as conducting vessels because they transport blood at high pressure to tissues. So arteries, blood flows under high pressure in arteries. They have stronger vessel walls and blood also flows at high velocity in arteries. Clearly, since they transport blood at high pressure to tissues, they are known as conducting vessels. So these 3 points in arteries. Arteries are the number first blood vessels that make up the circulatory system. Moving forward, the second blood vessels that make up the circulatory system is an arterioles. Arterioles. Clearly, 3 points in arterioles. These are last small branches of arterial system. Clearly, the arterial system in our body, these are its small and last branches. Clearly, small branches, and they act as control conduits through which blood is released into capillaries, and through these arterioles blood is released into capillaries. So what is the function of arterioles? They take the blood from arteries and give them to capillaries. Clearly, number second point, in arterioles they have strong muscular walls that can close arterioles completely or dilate the vessel several folds, thus having the capability of vastly altering blood flow in each tissue in response to tissue needs. It means that according to the tissue needs, arterioles can dilate or they can constrict, thus they can alter the blood flow to each tissue. Clearly, so arterioles have a capability of altering the blood flow to the tissues. So arterioles have the capability of altering the blood flow to the tissues. Why? Because they have strong muscular walls. Which can dilate or which can completely dilate, or they can constrict. Clearly, they can close or they can dilate. Number third point, in arterioles, they are also known as resistance vessels. Why are they known as resistance vessels? Because they offer high resistance to the flow of blood. Clearly, so these are the 2 blood vessels that make up the circulatory system. Now moving on to the third blood vessels. Clearly, the third blood vessel is a capillary. What is the function of capillary? They exchange fluids, nutrients, electrolytes, hormones, etc. between blood and interstitial fluid, therefore are called exchange vessels. So what is the function of capillaries? They exchange materials between blood and interstitial fluid, and for this reason they are called exchange vessels. Moving to the number point second, for this function of exchange, capillary walls are thin and have numerous minute capillary pores permeable to water and other small molecular substances. Clearly, that's 2 points in capillaries. So for the exchange function, capillaries walls need to be thin. They are thin and have numerous minute capillary pores. Clearly, now we have the next blood vessels. Next blood vessels that we have is a venule. What is the function of venule? They collect blood from the capillaries and gradually coalesce into progressively larger veins. So they take up the blood from capillaries and they progressively join to form larger veins. Now this is about the venules. Now the last blood vessel, that is veins. Clearly, so what is the function of veins? They transport blood from venules back to the heart. So they take the blood from venules and give the blood to heart. Clearly, they serve as a major reservoir of extra blood. So they serve as a major reservoir of extra blood and are called capacitance vessels. Since they act as a reservoir, so they are called as the capacitance vessels. Clearly, because the pressure in the venous system is very low, thus the venous walls are very thin. So low pressure, which implies venous walls are thin. So these are the 5 blood vessels that make up the circulatory system. So our question was, describe the characteristics of different blood vessels. So we described the characteristics. So what are these blood vessels? Artery, arterioles, capillary, venules, and veins. ठीक. Veins, they are called capacitance vessels. You know why? Capillaries are called exchange vessels. Similarly, arterioles, they are called resistance vessels, and arteries are called as conducting vessels. Clearly, these are the objectives. So moving forward, there are 2 more points to go. This table is given in Guyton. It's a very important table. So it's a vessel, and here is the cross-sectional area of that vessel in centimeter squares. Aorta has a cross-sectional area of 2.5 centimeter square. Similarly, arteries, arterioles, capillary, venules, veins, and vena cava. Clearly, now we have a relationship between cross-sectional area and the velocity of blood. Cross-sectional area and the velocity of blood. You know, V is the velocity, A is the cross-sectional area of that vessel. Clearly, so V is equal to F divided by A. V is equal to F divided by A. This is the relationship between the cross-sectional area and the velocity of blood. You have to remember one point. Since the same volume of blood flow must pass through each segment of the circulation each minute, since same amount of blood flows through each segment of circulations, which implies this F is always constant. This F is always constant under normal circumstances. So if this F is always constant, which implies V is inversely proportional to A. V is inversely proportional to A, which implies if A is more, V is less. And if A is less, V is more. What is V? Velocity of blood in a particular vessel. A is the cross-sectional area of that particular vessel. Clearly? Now we go back to the table. Artery have a cross-sectional area of 20 and veins have a cross-sectional area of 80. As we can see here, cross-sectional area is more in veins, which implies velocity is less in veins and velocity is more in arteries. Clearly, because V is inversely proportional to A. So velocity is more in arteries than veins because arteries have small cross-sectional area than veins. Clearly, also due to the low velocity of blood in veins can store blood. Clearly, so due to this low velocity, veins can store blood. Since veins can store blood, they are also known as capacitance vessels. Clearly, now from this table, as we can see, the cross-sectional area of capillaries is very high, 2500 centimeter square, which implies velocity will be very less in capillaries. So velocity of blood is very less in capillaries. That we can see here. Velocity is slow in capillary, which is necessary for exchange function of capillary. We previously we studied that capillaries are known as exchange vessels. Why are they known as exchange vessels? Because they exchange materials between blood and interstitial fluid. So to maintain the function of exchange, velocity is very slow in capillaries because cross-sectional area is very high. Similarly, 2 more points. Velocity of blood in aorta is 33 centimeter per second, and velocity of blood in capillary is 0.3 millimeter per second. As you can see, velocity is very less. You can correlate these velocities with cross-sectional areas from this table. So this is the aorta, 2.5, which implies velocity will be more, and capillaries cross-sectional area very high, which implies velocity will be very less in capillaries. Clearly? Moving forward, last point, length of capillary is only 0.3 to 1 millimeter. and blood remains in capillaries for 1 to 3 seconds. So length of capillary is only 0.3 to 1 millimeter, and blood remains in capillaries for 1 to 3 seconds. And in this time, in this very short period of time, 1 to 3 seconds, the function of exchange is done. The function of exchange is complete. The function of exchange of materials between blood and interstitial fluid. Clearly, so in this short period of time, this exchange function takes place. So this is all about our first question. That's this. If you get a question in this like this in your university examination, these are the notes. This is exactly how you will write it. Now get back to Guyton. 3 learning objectives. The next video is going to be about the learning objective number 2, then 3rd, and these 3 learning objectives are clear, which implies organization of the cardiovascular system is clear. Then after that, we will move on to the next chapter, that's properties of cardiac muscle. Then we will learn its learning objectives, and if we learn its learning objectives, then properties of cardiac muscle is clear. And by doing so and so and so and so on, we will complete the whole medical physiology Guyton and Hall. Clear? This is your Dr. Oogway. Share, subscribe, like, comment on this video clearly, and share this video with your mates. Love you all. Thanks very much.